Yes, sir. You are live, sir.
All right. Very good afternoon, everybody. Welcome to all the students, participants who are joining us today for the MOOC live session. Today, our theme is to look at proteomics and proteogenomics data analysis workflow. What I thought I'll start with giving you some flavor of how we utilize these workflow in our own research. So I'll take you to a very brief journey of one of the brain tumor which we work on, and this is the glioblastoma, very deadly brain tumor which we are working with Tata Memorial and other clinician hospitals. One of the major thing we realized, given the complexity of this tumor, understanding just the protein level information or proteome alone is not sufficient. So we should try to look at what happens, the changes at the genome level and the proteome level. Of course, one could integrate along with the transcriptomics and metabolomics to get more comprehensive picture. So proteogenomics aims to look at changes happening at both the level, looking at various type of mutations happening at the genome level, but how many of those mutations are actually responsible to give us the functional information at the proteome level, right? So this work is still currently under review, done by a recent PhD graduate, Ayushi Verma from our lab, in collaboration with Dr. Prashant from Karkinos earlier and now at Dattar Cancer Hospital, Dattar Genetics Lab. So this very good collaborative work gave us some very, novel picture of why we should be looking at this kind of integrated analysis. And that's I thought to give you at least a flavor of it. I don't want to make a research talk today, but idea was when we look at the Indian population of the high-grade glioma tumor, what we could see, like, many, this tumor has been already quite well studied in the Caucasian population, and the Cancer Genome Atlas, TCGA, has already given a lot of novel mutations that are happening in the patients, of the North American and European population. What we could see, like in Indian population, there are some very unique mutations happening with a very high frequency, like PABPC and CNDP1. Some of these are happening with the 85-90% high rate, and those were actually not reported in the previous TCGA data sets, right? So that was pretty interesting. We also looked at how different type of kinases and the entire proteome analysis is actually changing in the high-grade glioma as compared to the control or low-grade gliomas, general broad brain tumor. One mutation which is already well characterized in the case of the glioblastomas and the brain tumor in general is the IDH, isosaccharic dehydrogenase, as you know, is very important for the metabolism. IDH wild type and mutation, that is currently one of the standard for looking at the classification scheme of these tumors. So we also looked at how the different type of clusters are emerging based on looking at the IDH mutation. And what we try to integrate the mutation data along with the proteome data. And we could see there are three major clusters are emerging as a part of it, which was pretty interesting and novel. First time we could see that by integrating data, we are able to now look at the deregulated or dysregulated pathways, which are coming from different clusters, and each cluster is having a defined IDH kind of profile, like one cluster one is IDH mutant, while the cluster two is having the, this one cluster two is having the IDH wild type, and third is the mixed type. And each one of those are driving some unique biological pathways. So these are something which you can see the biology can be so much pronounced, so much enhanced if we are able to compile this information together. And then we are further looking at, like, how we can look at the impact of these mutation at the different type of protein level and what different clusters which we have identified, how the immune cell and other pathways are being mapped into these cluster type very uniquely. So again, this kind of a big project which is happening. But what I wanted to give you idea was essentially how proteogenomic analysis can give you some novel insight. Additionally, from our lab, we have developed some database and software, which I thought I'll flash here because we are at the end of this presentations and the MOOC live session for this course. So, and you are very computationally oriented group to work in the proteogenomic data analysis. So, Deepu Biswas, one of the PhD student earlier in the lab, currently a postdoc at Harvard, while working on the brain tumors, realized that there was not much normal brain proteome profile analysis. So we developed the human brain proteome map, followed by the brain phosphoproteome map. And then we have utilized this information to build a database along with pituitary proteome landscape as well. Then we realized that, okay, why not we utilize all the data which we have produced for different type of normal brain regions of proteomics and different type of other brain tumors and other kind of infection diseases, and utilize that to make a database. But then we also utilize the transcriptomics data, and we made the common data analysis workflows so that all the data which we generate or other people have generated can be followed with the uniform guideline, uniform analysis pipeline. So based on those pipeline, we have now reanalyzed all the data and put them in this particular database, BrainProt, which is live and currently available. The V3 version is running and recently published in the Journal of Proteome Research. I think I'd encourage many of you to utilize it and look at almost 56 brain diseases which we have, how to search for your proteins of interest and look at their expression profile, look at the good classified signals from that, look at their potential drug targets. All of that can be identified from the BrainProt. Additionally, we were working with Dr. Arun Vitta while I was at my sabbatical and UCSF. We were looking at how the cross-linking mass spec data can be much more efficiently analyzed, and we, as a team, together developed this Alpha Cross Excel software, which is recently published in the MCP and will be available for the users to now start using it. Idea is that we have used the AlphaFold in the backend so that any protein structure can be deduced. Using Prodi, we are able to calculate the residual distances, and using the PyMol, we can display the structural changes happening. Additionally, the follow-up work of the BrainProt and looking at the importance of how the proteins we can identify which are druggable targets. So Ankit Haldar, who is also your TA for Proteogenomics course, has done fantastic work along with the team of undergrads from IIT Bombay to develop this new tool, DrugProt AI, which is also currently published in the briefing in Bioinformatics Journal. Here, the idea was to look at entire human proteome of 20,273 reviewed protein, and of which look at maximum features, like most of the other prediction tools were using only few features of the protein, but here we looked at almost 183 features. And we took all the data from the FDA, which is for the approved drugs or the drugs which have not been approved or those which are under currently trial. So using all this data, then different type of machine learning models like random forest, XGBoost were utilized to make predictions for the proteins, how much druggability they can have. And as we build the models, more and more data was being released from the FDA, and we utilized our data, which was predicted data, and cross-checked that data with the FDA data and found out how much correlation we can see, like among the FDA approved and the pending approvals, or the they have said that this is not going to work out. So all those predictions we were able to map, and between 70 to 90% kind of accuracy we could obtain from our predictions and analysis. That became quite powerful. And think about how these computational analysis could help you to make better prediction and utilize the resources well to do the meaningful experiment. Otherwise, it will get failed at the clinical trial level. One could do much more computational prediction beforehand before you can start investing into the actual cell-based experiment. So I'm stopping here. I just thought to give you flavor of various ongoing activity in our lab, what we are doing, and in which way the different tools and software which we are talking in the course can be utilized by many of you, and try to obtain some meaningful information from that. In today's live session, our goal is to give you at least some more in-depth understanding of the different software tools and data analysis workflow which we are using for the proteomics and proteogenomics. And we have already given you, if you attended previous live session, a detailed sessions about the sample preparation, how to take your peptides and proteins and analyze those using the different mass spec analysis. We have shown you different metabolomic analysis as well, and we have talked to you about targeted analysis. So various type of workflows can be utilized. Now we are going to take those learning forward and going to give you some flavor of the NGS-based platform. Of course, NGS itself is a big field, and a lot is happening in that area. I don't think we are the expert of that field. Even for our proteogenomics project, we collaborated with the genomics experts. but we will give you at least some short flavor of the target gene sequencing, which we can do in our lab, followed by some of the protein microarray based work and the in general microarray data analysis workflow, which is another high throughput technology required for proteomics and proteogenomics. And then we will build further from it and talk to you about some of the pipeline for data analysis. So let me now hand over this thing to Ankit and Shashwati to start with the genomics analysis. Are we starting the genomics now? All right. So given the timing is a bit short, I think let's quickly first start with the microarray, directly the lab session. TS, please share from the lab directly the microarray based work.
My name is Vishali. I'm going to explain what microarray is. So microarray, in definition, is a high throughput technology where we can analyze vast range of biomolecules like protein, DNA, peptides, or metabolites like drugs, etc., on a very small scale. So this is a chip which is called a microarray chip. So basically, what we do is we immobilize different kind of biomolecules over here, proteins, peptides, or even DNA, and that is what the term it gives it to. Like if we mobilize protein, so it will be termed as protein microarray, and if we immobilize like genes, it will be termed as DNA. So this is what the chip we prepare, and we scan it, and then we do data analysis. Now let me go through step by step from sample preparation to slide preparation and how we do the data analysis. So first of all, this slide, we prepared this slide with, as you can see here. So we put a layer of our analyte. In this case, we put protein because let's start with specifying like a protein microarray. So we have immobilized a layer of protein on this slide, which obviously we cannot see over here because it's very on a small scale. And then we incubate with primary antibodies. So primary antibodies are present in our sample. For example, if we take blood sample, serum sample, etc. So those primary antibodies will be specific to these proteins or let's say antigens. It will bind to these antigens in a particular epitope, and that is how the incubation of primary antibody with the antigen happens. Now we'll incubate with secondary antibodies. Secondary antibodies, point to be noted here is that it is fluorescently linked. So these secondary antibodies will bind to these primary antibodies, and it will release a signal in the form of fluorescence. That fluorescence is what we need to detect here, and that intensity what we need to have for a data analysis. Now that we have prepared our slide, this is the scanner as you can see over here, and this is the region where we insert our slide. So now I'm going to show you how we insert our slide in the scanner. So this is the part where we have our sample prepared, and then we are going to invert it and we are going to put it in the scanner. Because let me just show it to you. So we have added our slide in the scanner. We'll close this. So what basically this scanner does, it uses a laser technology to scan the slide and tell us the intensity or that fluorescence intensity that we are getting for the fraction in this slide. So there are many different wavelengths where the intensity picks it up. For example, it takes the green laser for the 530 nanometer wavelength. It takes red laser similarly at 600 nanometer. So this so when we run our scanner and it scans the slide, this is the image that we get, as you can see over here. So let me just expand it. So these you can see are the spots. So these spots you see are the fluorescence detected when the secondary antibody binds to the primary antibody and gives us this signal. So this is the whole image. These are the blocks which are similarly proteins that are placed in this slide. So in total, in one chip, we have 14 blocks with same amount of, with same feature of same proteins. That means that we can analyze 14 different samples at one particular time. This is how we say it's a high throughput method or technology where we can analyze 14 samples at one single time, primarily. So let me zoom in on one block. We can see over here that these are the spots we can see, which shows that the binding has happened. So over here, you can see some spots are very brightly lit, some spots are blurred. So these brightly lit indicates that there is a very strong interaction of a secondary antibody with the primary antibody. So it shows that the protein that that is particular in that spot is present over there. And the blur spot that you see, they show that they have no interaction or very less interaction. That means no proteins are found or no interaction has been happened over there in that spot. Now, let me come to the data extraction part. So now that we have our scanned image with us, we want to know what are the intensities of these signals or spots that we see over. So there is a GAL file. So GAL file is basically a set of grid. So these are the small circles of grid that you see over here. Normally, our signal spots are called are given a term called feature, and these GAL spots, these small circular spots that you see over here, are called feature indicator. So what we do basically is we align these feature indicator with these spots. So you can see that I can move these circles. So what we do is we align these circles with the actual spots or the fluorescence that we see. So we align feature indicator with features to get to for the extraction of these intensity. So if you see, if I hover over any one particular spot, or let's say this spot, so you can see over here that at this particular wavelength, 532 particular wavelength, the F, which is the foreground intensity, is 3600, and the background intensity is 71. So this particular software, GenePix software, shows us the value of the intensity of the signals that we are getting from this whole chip or any block. Now we have our intensities. Now we want to check what the quality of the data that we have. So a microarray chip is designed in such a way that we have positive and negative controls in our slide layout. So as you can see over here, these brightly lit spots, these two spots over here and these two spots over here are the positive controls that we know previously when we get our slide or the slide is printed. So these indicate these positive controls, and just adjacent to this spot, we have these spots, these one, the second one, and over here, these are the negative control spots. Basically, we can use buffer or any compound. So it will show no fluorescence, no intensity from there. So we can, this is the first point of data check. So we can analyze from this that, okay, the data that we have are reliable. Second way of analyzing, second way of checking our data is that we can perform correlation plot. So what we do is we can perform intra-block correlation, or we can perform inter-block correlation. So let me explain it to you. So intra-block is correlation, which means that in a one single block, we want to check the intensity. We want to check the intensity of these data. So this particular chip is designed in such a way that we have the first row, second row as duplicates. So normally what we have in any chip, the similar spots, for example, protein A, is present in duplicates or triplicates to check the reliability, to check the correlation of these spots in that same particular block. So what we'll do the intra-block or inter-spot or inter-feature correlation to detect if we have reliable data. Also, what we can do is, if we zoom out, we have these similar proteins in the two blocks. So we can check the intensity as inter-block correlation, and we can perform a quality control check of our data. Now that we have done the data extraction and quality control check of our data, what we will do next is we will analyze those data that we have. So this software gives us a file called GPR file, which is gene fix result file. In that result file, we will have the intensity values of every feature of every protein that is immobilized, that shows the signal in every spot. So with that signal, with that values, we can analyze further our data using various tools, various programming Python like Python, R programming, and we can shape that data as per our project or as per our analysis or what we want to analyze. So basically, there are many wide, vast range of applications of microarrays, protein microarrays, DNA microarrays, and many more microarrays. So we can analyze function of any protein. We can analyze protein-protein interactions and many other, like we can also even see the gene expression. These studies can be performed using the microarray studies. So this is just a gist of microarray. It's actually a very mass and very emerging technology that we have, and it can be explored even more.
Thank you, Vaishali. So it was a good flavor of doing the protein microarray. I'll just flash one slide about where Vaishali left in terms of looking at these scanned images, what one could do from that, right? So there are different type of biological question you can try to address. In this case, you are looking at the brain tumor and the patients which are from different grades of the control to low grade, grade 1, 2, and 3, and the glioblastoma grade 4. All of those patients, almost 25 from each group we had. And we tried to look at from these patients' serum sample, whether we can obtain some early signature of antibody, which is known as autoantibody. And based on that, of course, the patient cohorts are very different. So you can see the intensities are very different across different groups. And so a lot of data analysis and looking at the data normalization was quite required before one could reach out to, to make any kind of meaningful sense of the data. But eventually we could identify 4 autoantibodies, which are very powerful signature for looking at progression of the disease, and one could predict quite early from the blood signature. So that was something interesting which came out of the protein microarray to look at autoantibody. Likewise, there are many applications which people are doing using these kind of array, and many times technology like mass specs are, while they're very strong and very robust, but they're not as helpful as microarray, because microarray you can directly take the crude serum and plasma sample, dilute them and probe on the chip, and you don't have to remove the one-end protein and look at other kind of interferences. So this is much more robust, and several applications you can only do on this kind of format, which you cannot do on the other technologies. In the meantime, let's just quickly flash through the genomics before we move on to the proteogenomic data analysis.
The instrument behind me is the ion torrent S5. This instrument detects the proton ions released during the formation of phosphodiester bond as a signal for sequencing. It comes with 2 other smaller instruments, which are ion one-touch 2 and ion enrichment system. The first step in a sequencing reaction is library preparation. Once that is done, we use the ion one-touch 2 system for clonal amplification of the library. After the library amplification, enrichment is performed on the ion enrichment system. It uses biotin and streptavidin interaction to select the template positive ISPs. The last step is to start the sequencing on the ion one-touch S5 instrument. For that, we load the sample onto the chip. The sample should be uniformly loaded on the chip, and then we place the chip on the chip clamp. We close the chip clamp, and then we check that the washing solution, cleaning solution, and reagent cartridges are in place. After that, we start the sequencing.
All right. So there are different type of technology in the NGS, which I'm sure like you have already gone through it. This is just one flavor of using the targeted sequencing, using ion torrent technology, what we are having. Next, we are going to talk to you about web-based gene set analysis using WebGestalt. And Ankit is going to give a session about data analysis using this software, which you have already got some exposure, but we will just refresh you and show you some application from that.
Yeah, hello everyone. Aware about just will refresh it once more. Yeah, so when we are talking about as Sarah has already shown that different proteogenomics analysis, so we do genomics, then we are getting protein, and we understand that there are different levels of mutations, followed by what we get the different protein expression and the different effects of gene changes and all are reflected in the protein. So now once we are aware that we have got the proteins, and we have done the secondary analysis. So I am hopeful that you all remember that you will get this kind of matrix where there will be protein IDs, you will get fold change, log to fold change, p values. So understand that okay, which of proteins are exactly your significant list of proteins, but then you need to make sense of it, like what you are want to what we can understand biologically, that what mechanism might have undergone that these proteins are coming as dysregulated, or these sets of proteins are coming as differentially expressed in the cohorts. So for that, with result, as you all know, and I think all of you have done some exercise as well, assignments as well. So this is the interface, and they have released the new versions 2024 apart from 2019 when they had done, but we were using we can use any of those, but again you can use the latest version as it would give you more features. So when we are understanding now, so you can see from the name of this thing is that translating the gene list into biological insights. So that definitely means that when we are doing, we are getting a set of a list of gene, a set of protein. But what is the actual biological insight that we want to do, and we want to counter, or we want to understand the mechanism so that we can develop new therapeutic targets, understand its insights, try to prevent the disease, or try to prevent the prognosis to aggressiveness, etc., that you can think of. So this toolkit helps us to understand that. So for that reason, we will do two analysis. There are three analysis you can do actually: overrepresentation analysis, gene set enrichment analysis, and network topology based analysis. So one major the network topology analysis is something which will give you the interacting networks, like this particular protein and this particular gene is connected to which proteins, or so that you can get a understanding that okay, so this is a protein that is your or a gene that you are finding it to be mutated or to be differentially regulated. So what is its influence, or what are its interaction partners, so that you can understand that okay, so how much it was connected and how much it is actually influencing its role in its disease. So you can think of a in an analogy, if a statement is made by any significant influential person, then the effect will be a huge to a large society. Whereas statements made by not so popular, or by you can understand by school children or by anyone else, a common man, so it will not influence a wide range of society. So that is the way to understand for the network based analysis, that how influential that particular protein is, or how much the spread is there, that okay, so this complete that whether it is a master or a key regulator and is influencing a variety of important reactions or mechanisms, like you can say of you can think of any like ECM receptor, focal adhesion, then you can have the EMT transitions. So how much influence that? Over representation analysis is something which gives us understanding that okay, we are having this particular pathways. These pathways So these many genes or proteins, the list that you are giving, so how many are actually being actually you are finding that, okay, is it mapping to the particular pathways already present in the database? So I will explain why I again go by step that your gene list will be actually is being run across or being referenced across a database from known literature, known biochemical studies, which is already present in the web. From there, it is being referenced into and will give you that how many entities are being actually mapping to your particular pathway and what is its enrichment ratio, what is its p-value, this kind of analysis. And gene set enrichment analysis is something where you need to give the complete your normalized data, and it will give you the understanding that which that the genes are making, which pathways are actually enriched in your cohort of studies that you are studying. So now we'll do overrepresentation analysis and network topology analysis and see the parameters and what does it mean and how we can do that. Okay. So here, for instance, when we are going to over-representation analysis, so first of all, you will be giving organism of interest. So here our organism of interest is Homo sapiens. But if someone does the experiments in any other organism, and for that different models are used for different kinds of validation and different kinds of analysis, then you can choose accordingly. So then you have to choose a functional database. So this is again very, very important. As I mentioned, that these gene insights, these cannot, the software or these toolkits are only helping us in curating from the huge reference literature and the database that has been worked around the years, around the globe by different scientists in different biochemical studies, functional assays, functional different pathways that have been known, that are present in the database. So this helps us in curating it us. So there are different databases, and now what we want to know actually, right? So there is a gene ontology, there's pathway, there's network. So what we want to know? So we want to know like pathway. Okay, so which pathways we can get? So these disrupted genes or proteins. So what are the basically pathways that are coming differentially regulated, right? So now these are the databases which contains of the different pathways like KEGG, Reactome, Wiki pathways, etc., etc. So these are all the functional. You can say that it contains all the information related to particular pathways, which genes are involved, which proteins are involved, etc., etc. Okay. So now we will add the KEGG. So now the analyte type you can use gene, protein, metabolite. If you are doing any kind of PTM analysis, etc., etc., you can give. So we are using gene or protein that we will add. You can ask, they are asking for either we can upload the list or we can paste it. So we will be just pasting our particular gene list. Okay. So from here, you can just go and you can just paste it. Okay. So you can have the different references. So we can have genome protein coding. Okay. So we are mapping it against the references which for the genome, which is actually protein coding, not the non-coding ones. Okay. Then you have the advanced parameters, like you can do different algorithms, number of analytes for the category, that 5 minimum should be there, or maximum. Multiple test adjustment we are doing, that is the FDR correction using Belgemini-Hochberg. So significant level you can give FDR. So you can 5% FDR, number of categories, etc., etc., and then we can submit. So we are keeping it mostly default and submit. Okay. So now it will run and it will give us like after, yeah. So see, it's quick and fast, and see what we are getting. So once we have given all the particular list of the proteins, so now it has given us the result, right? Like with the pathway list of the cake pathways, what are the pathways that it has actually got, and what is its enrichment ratio? So see, you can see thermogenesis and metabolic pathways are coming as your top. So you will see that what is our mapped input. So if you remember what I said, that whatever genes, the proteins that we are giving actually is mapped with the particular gene sets that are already present. So you can see that we have our input set, we have the total input set or total list of those for a particular pathway, and what is the overlap. On the basis of the overlap, you are saying that, okay, so this is highly enriched, or the p values, this depends on the actual number of entities that are mapping in your pathways. And just remember that here we are not considering the fold changes of the protein. So if you are doing for a gene set enrichment analysis and those kind of analysis, then the fold change or the normalized expression pattern or the expression values will also be considered, and that will actually take you, or that actually will lead you to understand whether it's a upregulated or down regulated. Here we can understand, okay, so this pathway is perturbed, and if the majority of the proteins are upregulated or downregulated in this particular pathway, on that basis we can understand, okay, so this is our this might be upregulated or downregulated. So you can see all these data, and then you can find out. So now after this, we will be now quickly doing another thing, is the network topology based analysis. Okay, so we have seen over representation analysis. You can just put up your own set of list, or you can download any data from your for any particular disease of your interest from your Proteome exchange and other, which have been discussed quite in a quite detailed way in your complete course. Then you can download, reanalyze, and upload, and can see that which pathway is actually it is getting mapped. So here you can see that it is Homo sapiens. Then you will be doing network, right? So there are different methods which we can use that, but we can use PPI biogrid, that is a protein-protein interaction biogrid. So because, as I mentioned, that we are looking at the network or the influence of the proteins with its interacting partners, and whether it is being interacted to a large scale, or it is influenced, or what is the way it is, or the how many proteins are getting interacted. So that we are adding. Again, we are adding the gene or protein. So here you can have different algorithms to add it, and you can have like the top 10, highlight the seeds and the neighbors, and then we can submit it. Okay, so ID type, again you can change. So since we are using, you can understand by the ID that we are using the UniProt ID. But if you are using a gene ID, that also you can do. If you are having a gene expression data, or a RNA-seq or expression data, so this kind of data if you have, so then you can also change it. Okay, so now you can add like UniProt, SwissProt. Then we can have this network expansion, then number of neighbors, then you can have like the different significance and the top ones if we want to see, and then we can submit. Okay, so this will give us again the network. So here what we are expecting, we are expecting a network. So network, you can it is based on the graph. So if you remember in the graph theories, that is unidirected graph or bidirected graph, both the graphs can be there. So similar thing happens in our biochemical pathways also, when we are looking at. Yeah, so now you can see that we are getting this pathway. So for example, you can have this particular understanding. Okay, so now you can see that the gene ontology of this particular is like mitochondrial transport, going with the NADH dehydrogenase, so enrichment, and the ancestor of enrichment. So not only which gene ontology you are getting actual enriched, but what is the uh, just ascending to it, right? Like protein containing complex, then you will get NDNADH dehydrogenase complex assembly, or mitochondrial respiratory chain. Then you have like the cellular component, protein containing component organization, Similarly, what you can do is that here, so again, you can download this also, and you can use for your different reports and different publications even. So you can see that if you just zoom into it, okay, so you will see that this is the network. So genes with the selected functions, right? So you can see this is the network where the different proteins are being, so you can see that whether it is connected. So now if you go downwards to interpret this particular, since it is quite complex picture, right? Complex image to understand. So this is the detailed information, right? So here you can get like which are the gene symbols, what is their random walk probability, what is the gene ontology ID, gene name. So you can have the number of genes in the category. So if you can see just right, so you will get that the number of genes in the category. So for again in the mitochondrial transmembrane, there are 58 in the category, but the input that we have given, that overlapping is only 14. Then you have the raw p-values and then the p-adjusted p-value or MDR. So similarly for mitochondrial organization, for mitochondrial transport, calcium import into the mitochondrial cellular. So these are the different pathways or the enriched gene ontology categories, and this is the protein-protein interaction map, and this is the number of overlapped genes with the categories and the p-values that you are getting. So this is another way, as I mentioned, that you were expecting in the network topology analysis is to understand and identify the different networks and how those kind of different networks can help us understand the gene ontology or the biological processes that are being altered or disrupted, and how much they are enriched or how much overlapping are there. And in this way you can decide on, and in this way you can understand, okay, so this is how you can finally lead to the that, okay, so how you can finally choose a particular pathway and try to inhibit that or understand the mechanism. So now here you have seen different pathways like KEGG, reactome. So then now I think after, so I would so request Amit to show the reactome that actually how the database is and the made, like you have seen that we are using it, but actually you can use those databases as well to understand and identify the different pathways. Yeah.
All right. Thank you, Ankit. So, yeah, I think, making the sense of the data is the most challenging part. While by now all of you will appreciate that a lot of data can be generated from the NGS platform, microarrays, mass spectrometers, right? But how to really think about what is the most meaningful biological information from that data set is always very challenging. So there is no one tool which we should rely on. We should try to make use of various type of pathways, network, different softwares and different resources to then try to see, are we reaching out to some core hubs which is actually helping us to understand what is driving that disease pathobiology, right? So now we are going to show you next good resource, very freely available, Reactome, which is also, I think, very good for the visual depictions of the pathways. Amit is going to take some data set from the mass spec and going to plot it and show you in the reactome.
This is Amit, and as Ankit has already showed you about the OFG salt, so I am going to.
Hello, am I audible?
Yes,
Yeah. So thank you. Good afternoon. Good afternoon, all. So I'm going to talk about the reactome and another software called network analysis. So as already Ankit has showed you that how we can take the data from an Excel sheet, and then we can use this particular UniProt analysis for further analysis. Now, so this is the Excel similar kind of Excel sheet that Ankit has already showed you. Now you can see the particular log FCs here. So see in this particular sheet, you can see there are a lot of upregulated and a lot of downregulated proteins are there. So now your objective is to find what are the different upregulated pathways and what are the key proteins that are associated with the pathways. So suppose from this data set, I want to see what are the different upregulated proteins are there. So I'll try to just copy these all entire UniProt IDs. So you can see I just stop here. So you can see from this Q9B, then this Q8N. So you can see a difference of 4 chains. So these are the proteins that are upregulated based on a log FC value. So basically we need to copy these particular UniProt IDs. Now, so now we have this Reactome database. You can just type reactome.org, and here you need to go to analysis tools. All right. So yeah, so you have your so you have the interface of the software. You basically need to just copy paste all the UniProt IDs. So now you will go to continue. So not just Proteomics, you can also use different kind of gene name or some metabolomics that you can also paste here. Now we will continue. So yeah, there are two important things here. One is the project to human as the input interactors. So project to human means suppose you are working with some other suppose you are working with some dengue or plasmodium samples. Okay, so on you want to see some proteins that are quite that are equivalent to humans or are kind of homologous to human. So that time you need to like take the project to human, and also if you also want to see the what are the interactors with our hits, you can also need to check this option. Now you go to analyze. Yeah. So this is the main. This is entire map you can see. So it's called the Voronoi visualization. What are all the other significant pathways that are coming in your data set? So these are the particular pathway names are there, and if you want to like go deep into the literature, what could be the potential explanation and what could the interactors, you can just click on it and you can explore it more. So yeah, so you can basically see here the entire pathway of this particular pathway is the post-translational modification. What are the key important features are there involved here? Similarly, you can also suppose you want to go for whole link diagonalization. You can also click there and you can have a detailed view that what are the important interactors or important UniProt IDs are there. Now, suppose you want you want to download the PDF file or the CSV file of your input of your output data that you have got from the Reactome. So you just go to downloads. So downloads are two things you can see. There's a CSV and there's an analysis report. So you just need to basically click here. The CSV file will get downloaded, and also the analysis reports. You need to just click here. So I have already downloaded that files actually. So I am going to just show you something on that. Yeah. So so this is basically the entire. Yeah. So this is basically the top one of the important pathways that are coming here. What could be the entities found and what is the entire pathway name? Most important is what is the p-value of these pathways? Are those pathways really statistically significant or not, and how these are correlating with the biology of our samples. Similarly, you can also see that what are the important proteins what are the important hits that came from our data sets and how those are actually associated with the pathways. So similarly, you can also see the pathway analysis report here. So here is the so here is the most significant amount of pathways. So there are total 8 hits that came into our data, and these are 84 means there are how many proteins are there in the background. So if you want to go to detail about all the pathways here, you can just read here and you can observe that what are the important interactors and what are the important pathways and what are the key important biology, and the data that is coming from this kind of analysis are really correlating with our disease or not. So this is for the Reactome, and the another software actually we use is the network analysis. It is kind of similar to the network and the WebGSalt. So basically, One of the important features of the network analysis that you can also just specify the organism here, and you can also specify what is the particular tissue specific regions also. So you need to just upload that. We have uploaded. Proceed. Suppose you want to see some protein-protein interaction here, you just add its tissue specific. Now, suppose you are dealing with some brain proteins here or brain cortex proteins, you need to specify the tissues here. You will click OK, and you will go to proceed. Then again, you go to proceed. Yeah, so this is the entire view of your network. So there are two important things here. One is the seed and another is the protein. So seed is basically the core of your molecular construct. And these yellow dots, you can see these are the interactant proteins. So similarly, you can use different database to find out what are the pathways here. Suppose I am here using a KIT pathway. You just click submit. And you can see what are the important pathways that are coming from our data set. So yeah, there are a lot of things, and you can also play with them and also can download and save your results. So that's
all right. Thank you, Amit, for showing the different ways of plotting the data, looking at pathways from Reactome and the analyst. There is another resource which is people use very often in UCSF over there is the enricher. So again, there could be different ways of looking at pathways, and I would suggest look at more than one. Also, I think what you guys will realize that when you're analyzing the data, many of your entities are also mapping like to multiple pathways, right? So then you should look at which of your identified proteins are mapping to which pathway. Then probably you are going to look at how best you can develop biology from that. So but finally, it's all about validation, because you have identified a lot of targets from your proteogenomic analysis. How to validate them, especially at the protein level, validations are very challenging given the need for antibodies. So alternative approaches to do that is to do the targeted proteomics using the parallel direction monitoring or multiple direction monitoring approaches. Shashwati is going to talk to you next about a software named Skyline, which can be utilized for doing targeted proteomic analysis, and how now you can take some of the meaningful protein which you identified from the pathway, then want to validate them further on the experiment. So Shashwati, over to you.
Thank you, sir. So good afternoon, everyone. So I'll show one software which you can use. It's a freely available software that you can download from the internet. So this is the Skyline software. You can download the Skyline daily beta version to utilize this. And another thing you will need for using the Skyline would be the background proteome. So for that, you can go to the UniProt, and then from there you can download as your choice. Like here, I'm showing for the Homo sapiens. You can do that for your choice of organism that you are working with. So you need to download the entire FASTA sequence from this UniProt. Then you can use the Skyline. So I'll just show the Skyline interface quickly. So there are three things that we need from the Skyline. First is the preparation of transition list. In the preparation of transition list, mainly we are showing that we need this particular protein of our interest just to monitor those proteins in the instrument, not the entire global proteome. So I'll just quickly flash through the settings that we usually use. So in the peptide settings, first you have to incorporate your database. In that way, it will know that which protein you are going to monitor. Let's say here human, I have already, it will take us some time if I'll do it here, but I have already incorporated the background proteome here, that is human. Or else you can use this add button to add any kind of database to the Skyline. Then you have to enforce each peptide uniqueness by its protein. That's because we don't want any repeated or sequence which will appear in our data. So always we need unique protein, that for unique peptide for that particular protein. After that, you can just use enzyme, like the enzyme you have used to do your sample preparation. So here we are using trypsin. That's why I have selected trypsin. Like that, all other tabs you can keep as the default. In the filter, we are monitoring mostly the peptides which has like minimum length of 8 and maximum length of 25 amino acids. Then you can use the library if you have already like run your sample in global. From that you can prepare your library as well. For now, I'm not using it. Then in the modification, you have to select the fixed modification and variable modification that as we use in the global setting. So here carbamidomethylation we are using for the fixed modification. Similarly, rest other things I'm keeping it by default. Then you can just click OK. Then the second settings is the transition settings. That also I'm just showing, but I'll keep all the things as a by default. So you can play around with the precursor charge, ion charge, or the ion types you want to monitor. So here also we can use the library. So instrument, all the full scan mode, that also we can set here. So for now, I am keeping everything as by default. So now I have to give my input protein list. So I have 4 input protein lists here. So you just have to copy it. Then from the edit section, we can go and insert the protein. So you can just have to copy and paste. Yeah, so now you can see 4 protein that I have already chosen. So you can just click on insert. So it will give me all the UD peptide of this particular 4 protein. So here in the left, you can see there are 4 protein and their unique peptides. So now I will export its transition list. So from the file, we can go to the import, and from there, export. There I can export the transition list. So transition list will be required for the MRM analysis. It will monitor each and every transition, but for the PRM experiment, we have to export the isolation list. So it will only monitor each and every precursor. So for now, I'm showing for the transition list. Here you have to select the instrument that you are going to use for the transition list. So here we are selecting Altis, where we usually do the MRM experiment. Then you have to set the parameter, and you have to click OK. Now I can save my transition list here. So after exporting the transition list, you have to run the sample on the mass spec on that particular transition list. So the instrument will only monitor those particular transition. It will not monitor other proteins present in the sample. So I will show how the result will look like after the analysis. So I'm showing one example here. Like I have already exported the result. So yeah, so you can see this is one of the peak of a particular protein peptide. So we then the second step is the refinement of the peak. That means not every peptide is going to come very good in the targeted experiment. So first you need to refine the list. So we have like lot many peptides here. So from that we have to choose which are coming good and which are coming bad. So you can see this is eluting at a very good peak shape. So this is the good peak. You have to keep it for your experiment. So the next peak you can see there is lots of background noise and they are there. So you can either delete some of the transition and it will appear good, or you can just delete this peptide. Similarly, I'm showing few of the results which are not very good. So you have to choose between them. Then you can refine your list. After that, in the refined list, you have to run your actual sample. So that in a way, you will save your time. And
Saraswati, thank you for a very quick overview, nicely for targeted proteomics. Again, students who want to really do this exercise, they have to spend more time on it, because this is just, one round. After that, you have to do a lot of refinement. You have to do rerun of the samples. Many things happens to make a good targeted assay. But major advantage here is we are not using antibody. We can directly validate the peptide using workflow from the either Orbitrap or the triple quad based mass spec. So just to close up today, I think many of you who have gone through almost 3 months, 12 weeks long course must be getting ready for your examinations, must be also apprehensive about what all going to happen in the exam. I think some queries were there in the forum on that part. So just to clarify that you have to watch all the video lecture, but we have already presented to you over the week's time, that should be clear to you. It will be pen paper-based exam, so you don't have to worry about the online analysis for any software for that matter. But we will probably show you some data set or some interpretation to be made from your observations for the given data set. So that I think your understanding should be clear about the output file of different analysis part, but you will not actually do the analysis in the exam. And but more than the exam, more than just kind of going through the this particular your test, what is more important is the learning from different workflow. And if you are able to get more comfortable into this area, you should be very soon be involved in many big projects where you can start utilizing your knowledge for the genomic and proteomics and proteogenomics integration, multi-omics integration. A lot of things can be done in this particular area. Very exciting space. Almost all the research in the biology and medical is now moving more and more towards this particular way of looking at multi-omics data integration. So I hope the course has given you at least the primer, the flavor, the excitement of looking at the big data in a different way. And hopefully many of you will continue your academic interest and your research in that particular space. Wish you all very good luck, and we'll be in touch for any query over the mail or in the forum. Okay, bye everybody, and thanks to all the TAs for doing the excellent work on the today's live session, as well as especially Ankit for giving all the good responses in the forum. Thank you very much, everybody.